So keep your questions handy. But I guess let's move right along to our next presenter. The next presenter here we have today is Teri Grout. Teri is the director and manager at the Alger Conservation District. Definitely does a lot of work for the SISMAs as well, does invasive species work and just general outreach to the Conservation District and all that good stuff too. So thanks a lot for being here, Teri. I'm going to turn it over to you. Whenever you can get your presentation up and going, you can take it away.
Okay, let's see if you'll let me share. Okay. Is my screen sharing enabled yet, Matt?
You should have screen sharing enabled, Teri, but
oh, there we go. Okay, and there it is.
Now I'll make sure that something's happening here. It looks like so far so good. If you put it in presentation mode, we'll make sure it's looking good on our end, Teri.
Okay.
Yep, there it is.
How's that?
Perfect.
Okay. And okay, it seems to be advancing and not advancing, so good. Can everybody hear me okay?
I'm hearing you just fine, Teri, and I'm seeing your slides go back and forth. I think we're in good shape.
Okay.
Thank
well,
you.
In that case, I'll get started, and then when I inevitably go over, it won't be quite as bad. Okay. Anyway, thank you all for being here. And I'm going to talk a little bit first about the biology identification, prevention, and a little less so about decontamination, since Elise has already covered a lot of that, although there are protocols specific to HWA decontamination that we'll go over at the end of this presentation. First of all, Hemlock woolly Adelgid. It's been in the United States for quite some time. It was discovered in Virginia in 1951. And it's spread pretty rapidly through the eastern United States to the point where it's killed hundreds of thousands of hemlock in the New England area and the Appalachians since then. The big danger around here is if it moves a little bit farther to the west. We have a huge hemlock resource, as you know, in the Upper Peninsula, and for it to get loose in the Upper Peninsula would be devastating to a great deal of our forests. Hemlock woolly Adelgid actually can take only a few years to kill a healthy hemlock tree, a large healthy hemlock, and it depends on the density of the infestation and the number of other stressors. But the fact is, 4 to 10 years is sometimes all it takes. There are 2 generations a year of HWA. The egg sacs that we will see in some of the images I'll bring up will hatch out in spring. And generally, there are a few winged adults as well as some wingless adults. The winged adults, fortunately, will only look for spruce trees to mate on, and the type of spruce that they need to mate are not endemic to the United States. So these particular guys, the wingless adults, will not be able to reproduce. They'll just die without reproducing. However, the wingless females can reproduce parthenogenetically. Parthenogenesis, great word. Basically, they produce asexually. And these guys will then go through 6 stages of development, including an egg, 4 nymph phases, and adult. So the main thing is that with that many females, even with the wingless or the winged ones dying off, you could still have some pretty phenomenal amounts of reproduction based on what they can do. Each of the small white masses that you'll see can produce about 300 adults. So once again, it doesn't take much for these little guys to get a foothold once they get established. So, and there's the little egg sacs that I was talking about previously. So HWA kills hemlocks by feeding on sap from the hemlock shoots, basically depriving it of water and nutrients. And the fact that they have to inject into a hemlock twig means that you're not going to see them on hemlock needles. HWA is present on the hemlock twigs on the shoots exclusively. Those little white cottony looking sacs are pretty obvious and are probably more obvious to the naked eye than any other phase of the HWA life cycle. So when you're looking for it, you're going to look for white cottony sacs on the stem, not on the needles, and generally at the base of the needles, right on the stems. Now, once an infestation has reached critical mass, you'll also see decline in the hemlock itself. So, if you start seeing needle loss, gray-green crown, a thinning of the crown, and dieback of twigs and branches, you might start to suspect HWA. It could certainly be other hemlock infestors, but that might be your first clue that a very large hemlock is infested. The best time to look for it is during leaf off, which is why we do HWA surveys from about November till April. The fact is that even with leaf off being in full swing in the winter, you're really not going to see much except those white woolly egg sacs. And in a tall hemlock, you might not be able to see much at all. So, we are looking for that phase, that hemlock woolly adelgid egg sac. And the reason we don't do surveys later in the year is that after about April or May, the larval instars could be active. These crawlers, these larvae, are very small also, as you'll see in some future slides, and very easily transported. Like all the other invasive species that Elise just talked about, really things are so small and so easy to transport that prevention, while being the best method of controlling them, prevention is very difficult when you can't even see what you might be transporting. This slide shows what an extensive infestation looks like. This is a photo from the Appalachians, and you can see the gray-green cast of the dead and dying trees. It's a pretty scary sight and one we don't want to see happen in the UP. So one question that we have here in the UP is whether cold weather would actually prevent the viability of hemlock woolly adelgid. Around here, we say 40 below keeps the riffraff out. And it's possible that the extreme cold temperatures that we usually have in the UP might not allow for reproduction of HWA. There might be a northern boundary beyond which HWA can't be viable. There is some evidence of winter kill. Connecticut has done some experimentation on that, and it says that -4 or -5 degrees Fahrenheit might actually prevent HWA from being viable. But unless it completely wipes out an infestation, it can rebound again if you have a warmer year. And what about the insulating effects of snow cover? Here in the UP, we've had some pretty cold winters, but last winter was not one of them. While at this point we don't have HWA in the UP, at least not that we have discovered so far, we didn't have very much snow cover, and we probably didn't get below zero for more than a few days, even in Chatham. It's highly unusual, but it also could mean that if HWA is present, that potential winter kill would not be happening. So it's hard to say what the mild winter might have done. So let's go on to the identification. Once again, I think everybody's seen these little photos of tiny fuzzy egg sacs, but we'll go into a little more depth here. So first, find the HWA in this photo. Too bad I can't have hands raised. In a sparse infestation or a brand new infestation, you might not have very many egg sacs that are visible. On this particular one, on the little twig on the right-hand side, second from the top, there is a single egg sac in the axil of the twig. That kind of thing is not going to be very obvious. And I think if you did discover HWA by finding that one egg sac, my hat would be off to you. Usually we don't discover them until much farther along when the twigs and the stems are completely covered with fuzzy egg sacs. More like that. Now this is a photo of what you might see later in the season. The egg sacs, when they're freshly woven, they actually are a pretty bright white. They're kind of easy to see. But once they've weathered for a year, you know, the eggs may long be gone, but you'll still see the little fuzzy remnant egg sacs in a really nondescript gray color. These are really hard to pick out, and it's why another reason, one of many reasons, we don't do surveys in the summer. You're just not going to be able to see the older, shredded, degraded gray egg sacs as much as you would the brand new ones that are laid or are covering eggs that have just been laid. So it's pretty ratty looking. You might still detect it. It pays to keep your eye open, but really that's not when they're the most visible. It's really hard to detect those grayish tatters. Okay, and here we go. What do we have here? We've got crawlers. Really hard to see. And that penny will give you a little perspective on just how bad they are and how difficult they are to see. But, you know, when these larvae look for feeding sites, they pretty much insert their mouth parts into the stem, and guaranteed once they are on the stem, you're not going to see them. Now, crawlers, when they are present in summer, they estivate. That is, you can see on this photo, there's kind of a halo-like little bit of fuzz around some of these crawlers. They're still very small. They're really difficult to see regardless, but that halo, that ring of wool, can also be an indicator if you happen to see something embedded on a stem. The Adelgid hatches from eggs into this crawler stage, and then they will, then they can form this estivating ring of wool. But it's still best to consider the whole summer period as a time when the crawlers could be mobile, and you should definitely avoid transporting hemlock material. You should definitely avoid especially transporting that in an area where there may be HWA. And now we'll go into a little bit of identifying what isn't HWA. You know, the photo on the left, Hemlock woolly adelgid itself, yeah, those are pretty obvious. You've got little egg sacs up and down the stems. That is the classic photo of what it looks like. One look-alike that we actually, I don't think, have found in the UP yet is elongate hemlock scale, or EHS. The hemlock scale actually creates this waxy secretion, and it can build into a mass that kind of looks like a fuzz on the base of the needles. But again, if there's fuzz on the base or on the needles themselves, it's not HWA. It might not be EHS either. It's kind of hard to tell. But at any rate, if you're seeing it on the needles, it's not HWA. Now, there are other pine scales, other armored scales that you might see on the underside of hemlock needles too. So EHS can be misidentified. It can be a little bit tricky to identify them, and I owe a lot to Simeon for pointing out the difference here, because we actually thought we had found EHS multiple times last season, but what we were probably looking at was a different pine scale species. If you look at the photo on the left, if you see it's a little bit misshapen, this is probably a pine needle scale, and if you see the I guess it's what's called an oyster shell shape, where it kind of tapers down to a little point at the tip. That is probably just a pine scale. The photo on the right, this is some elongate hemlock scale from the Lower Peninsula. And if you could see, this scale is actually pretty long, you know, and it occurs on the needles, as I mentioned. You can sometimes differentiate the species by the color. The female scale is going to be kind of brown and tan, and you can see a lot of females in this particular photo. There are some males on there that are way harder to see. They have more of a white covering on them, and it's very hard to pick that out in this photo. But either way, both of them are longer, and that armoring, that casing, is probably going to be kind of a brownish tan, especially in the females. The main thing with elongate hemlock scale is we're not really sure how much damage it does to hemlocks, but we have noticed that it can occur in concert with HWA in places in the Lower Peninsula. So, whether there's a connection, whether, the two diseases or the two infestations can feed off each other, whether EHS actually can cause some damage when it occurs in conjunction with another problem, that's kind of hard to say. However, at this point, we are observing, and we will report any EHS that we find during our Hemlock surveys. And we'll also hopefully define that it's not a big problem. Maybe we won't find any in the UP at all. Okay, there's a close-up of some HWA ovisacs on the left-hand side there, and you can kind of see some little crawlers there. Again, pretty obvious, pretty fuzzy. On the right-hand side is something that's kind of fuzzy too, but spider egg sacs, like the one on the photo in the right, can often be mistaken for Hemlock woolly adelgid. One thing that you'll usually see with something like a spider egg sac is that there are a lot more strands coming off of the sac. It's not such a compact little ball of fuzz as HWA ovisacs are. Now it pays to be cautious about this. So if you see something like that could be HWA, even if it's got some little strands hanging off of it that could be spider webs, it's best to collect and have it identified. These egg sacs, may not be clinging as tightly to the twigs as you would see with HWA. Maybe they're mounted on a needle, but nonetheless, they can resemble it very strongly. So it pays to be cautious with that and to observe carefully or take a sample. If you do take a sample, I'll have some information later on in the presentation on how to submit it safely and legally for identification. HWA also can cause damage to and yellowing and browning of the needles, and you may see that as the infestation progresses. So if you're seeing that needle dieback, needle drop that I mentioned, or a thinning of the crown characterized by that kind of needle dieback, you might want to examine more carefully for HWA egg sacs. However, other HWA lookalikes can also cause browning of the needles, and one example is the needle miner on the left, which is, actually mining the needles, sucking the juices out of the needles. That kind of damage is not terribly, detrimental to a hemlock, but again, it can be mistaken for some of the needle problems that you would have when there's an advanced HWA infestation. And, once again, we're looking at hemlock HWA drying and graying of needles. On the right side, you might have a dying back of needles or browning of needles caused by spider mites, which also could pierce plant cells and suck out the contents. So once again, every needle browning is not something to be worried about, but it usually bears a little closer look, And around here, since we're trying to identify HWA if it exists in the UP before it becomes a major problem, I'd always prefer that you were cautious. All right, more HWA lookalikes. Again, the classic HWA pattern of ovisacs on the left. But the oak skeletonizer can also occur throughout the Great Lakes region. And these feed on oak trees, but they can also feed on other needles if they choose. Again, needles by themselves are not a good indicator necessarily of HWA. Okay, and mealybugs. This is one that can be a little bit confusing too. So once again, classic HWA that we have looked at about a dozen times so far. But on the right-hand side, we're looking at mealybugs. Now, mealybugs, it's a common name for a huge family of scale insects, Pseudococcidae, actually, if you really care about that. And these are typically agricultural pests. We don't typically see them causing a lot of problems in forest settings. However, even though they're not really considered a threat to hemlock, you could see how those little mealybug egg sacs could be mistaken for HWA. Kind of interesting, though, mealybugs actually get farmed like ants, by ants, much like aphids do. So kind of fascinating creatures, and you could find some mealybug sacs on hemlocks, but since they can be confused with HWA, be a good idea to take photos, to report them, or to take a sample if you find them. However, if it does turn out to be mealybugs, anything in that Pseudococcidae family, don't worry about it. And of course, there's the other imposters. You know, people have confused dryer lint and dog hair and hydroseed mulch and drops of pine sap and all kinds of other things for HWA. Again, when you're looking at something that small, something where you can't really identify the insect by eye, and where you're just looking at the egg sacs, where you're looking at the other things to indicate the presence of the insect, there could be a lot of things that could be confusing. It's not a super obvious thing like oak wilt with, you know, a severe leaf drop and dieback happening as fast as that particular problem does. So how do you prevent HWA spread? Well, as I mentioned, Hemlock woolly adelgid can't really fly. Again, not counting those few winged females that emerge and are not able to reproduce. But those crawlers, as you saw in the previous photos that I showed, are tiny. They can be transported on the feet of birds. They can be transported by wind, by animals. In fact, those little sticky egg sacs can be just sticky enough that they can adhere to wings or to other animals and be transported full of eggs. But one of the other main problems that we have is human activity. Yeah, we travel pretty far, and we do have the ability to transport crawlers and infested material. We also sometimes are careless about how we dispose of things. You know, downstate in the counties that have Hemlock woolly adelgid, you know, moving, throwing away, you know, or improperly storing or transporting infested hemlock material can be a huge risk. And of course, don't move firewood is always a good idea too. But the highest risk period is during those spring and summer months when the eggs and crawlers are present. So we really have to worry about that period. And unfortunately, that also happens to be the period of time when people are coming up to their camps, traveling for the summer. And if they are coming from an area that has active HWA infestations, that can be a problem. Prevention is always cheaper and easier than getting rid of an infestation. So specifics on HWA decontamination, besides the things that Elise discussed earlier, that is not moving you know, anything in your car, in your boots, or on your equipment, we do look at minimizing contact with infested hemlock during that April 1st through July 31st timeframe. We advise to park and drive vehicles away from hemlock limbs just in case the hemlocks that you happen to be parking under might be infested. We do advise cleaning contaminated equipment between sites in the way that Elise went over in her presentation. And during the high risk period, especially if you are anywhere in the 5 counties in the western lower peninsula where Hemlock woolly adelgid has been found, we recommend cleaning boots and washing hands with soap and water or hand sanitizer between sites. We recommend that any equipment or clothing that may have been contaminated by crawlers, which again, you're not likely to see, be either sealed into plastic bags before you go into an uninfested area, and or washed and laundered before wearing again. We're not worrying about that so much here at this point, but we are exercising caution and recommending washing or drying on high heat any clothing that we wear in dense hemlock stands, just in case HWA is there, and just in case it's, you know, we've missed it. Any pruners that have been in contact with HWA or any other equipment should be wiped down with a bleach, 10% bleach solution in soapy water before moving to the next site, or putting them in a, you know, a dirty or contaminated bin until they can be sanitized. So this is the end of part 1. I'm going to put up another little presentation to take up the next few minutes about the HWA survey program in the UP. So I guess if there are any questions on this, if you want to ask them now, great, or if you want to save them till I get through my next 10 minutes, that would be great. I'm going to stop share and put the next one up, Matt. Good?
Yep, that sounds good, Terry. I'll monitor the chat room for any questions, or anyone can feel free to chime in if they want to ask a live question of Terry.
Okay. And I had the thing up. I'm looking for it. Hang on just a second.
Yep, take your time.
Okay. Oops. Let's see. Ah, there it is. Okay.
I think it's working.
All right, good. And this one's moving too. Okay. All right.
Looks good, Terry.
Okay, so far so good. All right, so I'm going to talk a little bit more about the survey efforts in the UP and the survey and treatment response in the Lower Peninsula. So the first thing is, what exactly does our survey program do and what doesn't it do? First thing is, this is not designed to find that one fuzzy egg sac at the top of a 100-foot hemlock. That first slide I showed you with one single egg sac in the axil of a hemlock twig, you're not necessarily going to be able to see that. Again, if you do, kudos, I bow down before you, you got eagle eyes. But again, looking at that little egg sac on the left, you won't see the individual eggs, but you do see the fuzz. Now the surveys that we're doing, we're really designing them to cover a lot of ground quickly. The goal is to discover whether we have a large infestation already existing in the UP. If we already have an infestation similar to what they found downstate, we do need to find it and act on it as quickly as possible. We can't necessarily assume that because we haven't seen it yet that it doesn't exist, and that's what our surveys are designed to find. The survey techniques I'm going to go over seem really basic, almost too quick and dirty to be effective, but statistics show it has a high probability of discovering an established infestation if that infestation exists. So at any rate, we are focusing primarily on the Lake Michigan shoreline up to about 5 miles in. And the reason for that is that, we have a lot of transportation that is possible by avian vectors, that is birds. And frankly, if they've been traveling from the Charlevoix area and the western part of the Lower Peninsula, they can have brought those crawlers, those egg sacs or something else over, and they're most likely to alight somewhere along the flight path, possibly nearest the shoreline first. So survey technique, when we go out to survey, we establish a name for a site, we take basic acreage and location info, and we'll survey everything from a single large site, for instance, 320 acres of lime forest land, or a large hunting campsite. And we'll also look at smaller residential sites along that area. Basically, if it's got hemlock, even only 1 or 2, we want to get a look at it. And then we traverse the site. We try to be systematic. We'll often start with obvious vector areas, following roads and trails if they exist. And that's for the same reason that Elise talked about, that is, humans and animals use these trails and they can be a major mode of transport. Then we try to check up to 30 trees per acre. In some areas with very dense hemlock, that might be only a fraction of the hemlock that exists on a site. But again, if we are spreading out these 30 trees over the entire approximate acre, we're probably going to get a pretty good idea of whether any trees there have a heavy infestation. So by doing that, we should be able to catch those established infestations early. Then we actually examine the trees. Because HWA occurs primarily on the underside of the branches, we'll kind of flip branches up, check the undersides of the shoots if we can see them. In some cases, we'll have to use binoculars. In other cases, we may have to use pole pruners to take a small branch out to get a closer look. That's also the way in which we would take a sample if we felt we had a suspicious branch and needed to send that in. We try to look at 4 branches per tree, 1 on each of the 4 sides. And in some cases, as I've told you, we need to use the tools. Pole pruners are great for doing that. Binoculars are good too. Tall people are even better. And in some cases, the UP snow load is a real benefit. Not so much this year, but in other years, yeah, it was really nice to be about 3 feet higher up on the snowpack. And for the ones that are out of reach, we can try using binoculars. We can also look at any downed branches on the ground that may or may not have come from that tree. And in some cases, we'll look at smaller or younger hemlocks as they can be infested too. You know, again, it's imperfect, but we are trying to get a look at enough hemlocks within each acre that we have a pretty good idea whether an infestation already exists. But in the cases too of the larger hemlocks where you just can't see the underside of the branches, that's a good argument for stepping back and taking a look at the crown of the tree, looking at the condition, observing any yellowing or top decline, and maybe if you do see that, look more closely at the smaller hemlocks and any branches that you can observe. Okay, and how do we select the sites? Well, this is actually last year's photo, but this was a survey that we did at Harlow Park in Marquette. Why would we pick that site? Well, it's heavily used by hunters, hikers. There have been recent trail construction activities, which of course could have brought in HWA, you know, during the construction phase on equipment. So that we consider that a high-risk category. This was actually taken last November before all the snow hit, so definitely not a typical UP winter. And anyway, we also look at whether, you know, people and other animals can spread those egg masses, and that can be humans and their equipment, or it can be animals. And since birds are considered a possible means of transport, we also look at major avian flyways, migratory patterns, chains of lakes, which might be favored stops and flyways for migrating birds. So these are all priorities when we decide what sites to look at. We also look at sites based on ease of access. Earlier in November, before the snow hits, we'll try to get to those remote sites that we will not be able to get to later in the season if we have a normal UP winter. This winter was pretty easy though, so I probably tempting fate by saying it, but boy, what a nice season to survey in. So what other conditions make a site high risk? Well, major waterways, again, partly because animals use that, wildlife corridors, avian flyways, people. We look at lake shorelines and river corridors. Again, people are a big thing. We look at brush dumps. You know, we've had a lot of people observe that brush can be dumped by people who, you know, just clear it out of their area and take it out to the woods. And that, unfortunately, can spread a whole range of invasive species, not just HWA, but it can be a real risk factor. Anytime you have a community compost site or an illegal brush dump, you have a high risk of something being transported, HWA or God knows what else. Campgrounds are huge, you know, roadways, trailheads, and trails are big. Old trees and large mature hemlock stands are targeted for our surveys just because of the risk to these established hemlock forests. High income areas are kind of a risk factor too. It's suspected that some of the HWA downstate might have been brought in on large balled and burlap nursery stock by people who can afford to plant big balled and burlap nursery stock. And while there are quarantines in place to prevent the transfer and transportation of contaminated hemlock, that doesn't necessarily mean that something large could not have been planted as a landscaping plant already and started an infestation. You know, we look at forest edges, not just because of the easy access for people, but because edges are often used by many species of birds and many species of wildlife. And again, if they are in a position to transport that, that's how that can come in. We have also been taking a very careful look. Allegan, Ottawa, Muskegon, Oceana, and Mason counties are where the Hemlock woolly adelgid already exists in the Lower Peninsula. So we have been paying attention to our ownership lists from the county equalization websites, and folks who have primary residences down in those counties, their second homes up here are high on the list for doing hemlock surveys, just because of the risk of transport. How do we collect and verify data? Moving right along here. We've been using an ArcGIS app, Survey 123, using an iPad. We enter all the data that we talked about earlier. We record whether HWA is there. We record other things like approximately how many hemlocks are in the area, how many trees we surveyed within a particular site, whether we found anything, whether we found EHS, which we thought we had, but we had not, whether we collected a sample, and we take photos if we can. Now, here's the big thing. If you suspect you have found Hemlock woolly adelgid, here's the procedure for reporting and or collecting. The first thing is, don't move the thing unless you are able to properly bag it. If you found suspected HWA, leave it on site. Take your data, take photos, take coordinates if you can. Do not publicly announce that you may have found it. We do want to wait for this to be confirmed by MDARD or by APHIS, the Animal Plant Health Inspection Service arm of the USDA, before we go spreading the word that we may have HWA up here. As you've seen, there are plenty of things that maybe aren't HWA. But at any rate, the main thing is to make sure that whatever you detected can be found again. So if you can collect GPS coordinates, please do so. If you can take photos, and if they're geotagged, so much the better, please do that. In some cases, it might be possible to rule something out strictly on the basis of photos, but that's a good first step. If the branch is still attached to the tree, flag it. You want to be able to locate this again. If you happen to find a branch on the ground that's a suspect, please flag the tree that you suspect might have been the source of that, and mark with flagging tape any portion of the branch that you don't submit as a sample. Now, if you do have a suspect sample, before submitting, you need to have a compliance agreement from MDARD in order to ship these samples for identification. I suspect many of you probably don't have that. Alger Conservation District does have an MDARD compliance agreement. So if you find something, the easiest thing might be to contact us and arrange for us to get the sample, pick it up, bag it, label it, and submit it ourselves. If you do decide to take a sample and submit it yourself, though, please make sure that it is completely bagged and enclosed. So at the location, not after moving off, double bag the sample using plastic bags, seal it tightly any way you can. You want these to be 2 airtight bags, one within the other. Before shipping it, make sure that it's packaged so that it'll be secure while in the mail or when you get it to us. And make sure you've got information, the latitude and longitude coordinates, first and last name of the person submitting it, contact information, email or phone, who you're with, the date of collection, and if you have been IDing the site in some way, some other identification for the site. At any rate, once again, contact us if there's any question about how to safely submit this. Now, when HWA is confirmed, again, I'm kind of hoping it's not in the UP. It's not applicable here yet, but it might be in the future. In that case, we start doing delimitation surveys, focusing on high priority areas and looking at the northernmost infestation areas. At this point, HWA kind of is isolated in some of the sites on which it's been found in the lower peninsula. It's bounded on the west side by Lake Michigan. And a lot of the areas on the southern boundaries of areas that have found HWA infestations don't have a lot of hemlock. So to some degree, the infestations are kind of naturally blocked off. However, on the northernmost areas where HWA has been discovered in the lower peninsula, those are a little more closely connected and contiguous to hemlock resources. So we're really trying to be sure that this doesn't spread any farther to the north. Anyway, delimitation surveys are going to start at the infested trees and work outward in all directions until you can no longer find an infestation. So all hemlock trees within 800 feet of a confirmed HWA infestation are going to be treated. If a positive tree is found, then basically they're going to be treated whether or not you find HWA on them. Because we're going to assume that if HWA is that prevalent on that first tree, that there's probably at least a few egg sacs on the other trees. So delimitation data, a lot of similar data to what we collect, and I'm going to just speed through that. But treatment options, when it's confirmed, are what I want to talk about briefly at this point. So there are a couple chemical options. Imidacloprid, that can last quite a while. You apply it as a soil drench or injection, and you can apply it as timed release tablets below the soil line, but you can treat quite a few trees per year because it's going to last a little bit longer. Dinotopherin is water soluble and it moves faster, so it protects the tree more quickly, but it only lasts 1, maybe 2 years if you're lucky. Can do a soil drench. You can use it as a basal spray. And whether or not elongate hemlock scale is a serious problem for hemlocks, the bonus is dinotopherin also kills that. But the main thing is both of these are neonicotinoids. They can be extremely toxic to pollinators, and they can be very persistent in the soil. Yeah, well, you know, they last for several years, so yes, they're persistent. The residues in the plants can kill pollinators long after this has been applied. The good thing is the hemlocks are a wind-pollinated species, so really pollinators are not likely to be present near hemlock stands. But still, if there's significant, you know, floral blooms, native wildflowers or anything like that near a stand to be treated, you need to be very aware of that. You need to be very aware of drift watch registrants. You need to be very aware of annual application limits. We really don't want to cause a huge problem by being careless. And labels vary, but you also have to be very careful about the ones that prohibit application near waterways, and many of those have a 75-foot buffer around waterways. So there's not a lot of alternative treatments. You can do some basal bark treatments. Pyrethroids, insecticides that are more commonly used to prevent insect stings on animals, can also be used, but they have proven to be very much less effective than the neonics that I mentioned earlier. They will not be able to penetrate that waxy ovisac and get to the eggs. So it's, yeah, there may be alternatives, but we don't know that they're good ones. So I'm just going to run through a couple of photos of guys treating hemlocks downstate. Again, haven't had to do that in the UP yet. We also do have some examples of some of the products that are available that can be recommended to homeowners, but you know, we're not specifically recommending these brands. We're just showing this as an example of what can be available. And where are we now? Well, here are the surveys that we have as of April 30th, 2020. I'm right now in the process of collecting data from the other conservation districts and SISMAs that are doing the surveys from Sault Ste. Marie down to Menominee. And I expect to have that data within a few days and we can get everything updated. However, the surveys that we do are automatically updated to the HWA viewer, so those are going to be visible on that website. We're also looking at Hemlock woolly adelgid survey and treatment in state parks. That's being conducted by the DNR. This is, as of February 2020, known HWA sites downstate. You can see that we have, you know, quite a number of those, mostly along the lakeshore and a few farther inland. The most recent infestation discovered was in Ludington State Park, northern Mason County, and that actually was found last October in an area that had been inspected before. So just because you've been through once doesn't mean you can take your eyes off of anything. Yeah, we can't rest. But we are looking at trying to stop those infestations before they get to significant stands of hemlock in the northern lower or in the upper peninsula. There are some quarantines available, and those are updated as they change on the MDARD website. I've put down the MDARD contacts, Robert Miller and Mike Bryan, who can give you more information on that. And the information is also available on the State of Michigan website. That's kind of all I had, and I realize I've gone a bit over, so I appreciate your patience. I guess, I don't know if we have time for questions, Matt?
Yeah, absolutely. We definitely have some time for questions. You ran a little bit over, Terry, but like I said, my segment is going to be pretty short, and we'll be able to wrap up just after 4 p.m. here for sure.
Oh,
So yeah, we'll open the floor to any questions for Terry Grout, please, or at least field some questions too, if there's any out there. So type it in the chat box or unmute yourself and ask questions, folks, if you have them.
Terry, this is Warren again.
hi.
What about these Adelgids on balsam and on European larch?
Okay, so there is balsam woolly adelgid, you know, and I don't know that we've discovered that in the UP yet. Matt or Simeon might have a better idea, but that is a distinct species, although it very much resembles HWA. So I don't actually know nearly as much about those as I do about HWA. Kind of shows you where my focus is. But Matt or Simeon, do you have anything on that?
Not myself. I really don't have anything on the balsam adelgid. I do know I've seen the beach woolly adelgid out there. It was several years ago though, and that's common, but it's not very destructive. And it's a much fuzzier insect that kind of actually shakes when you get close to it. But I guess I would defer to Simeon if he has anything to chime in.
Yeah, the balsam woolly adelgid, something we're on the lookout for as well. We did put an article in that Forest Health Highlights this year. It has not been detected in Michigan, but you know, again, it could get to you either on holiday greenery or nursery stock or something like that. It is present in other areas of the northeastern U.S. And yeah, it would be a concern for our balsam fir. And it causes what we call twig gouting, or these swollen twigs on the trees. So you're not going to have the little white tufts right at the base of the needle. Have the adelgids just right along the bark on the branches, but more obvious would be the swollen twigs that it was. And there is some images of that in our Forest Health Highlights.
Fabulous. Thank you, Simeon.
Hey, Simeon, actually, is there a quarantine on any balsam products because of balsam woolly adelgid?
Yeah, actually, that's a good point. I think there is. I'm not remembering the details on that quarantine at the moment. I should probably look that up again. They're just kind of escaping my mind, but I think there is a quarantine from certain areas in the Northeast, if I remember correctly.
Okay. Yeah, something to look into. Okay.
Any other questions? Go ahead and chime in if you have a question. Okay, well, not hearing many right now. Of course, the chat window will be open for a few more minutes, and there's emails here for everyone who was a presenter today, or you can find their contact information through me. So if something comes up you want to ask, just definitely get a hold of one of us.